Today we're talking about species and speciation, the origin of the species and the biological species concept. So you guys have all heard the term species, right? And you use it broadly. This is a species, that is a species, this is not the same species as that. You are a different species than your dog is. They're different. You know that. But it's a lot more complex than that, and we'll end up finding that a species isn't so readily defined. We love to classify things. Historically, humans are all about classifying. We have this innate need to sort things out. These are all different. We can look at them and be like, yes, a turtle is different than a dolphin, is different than a bird, is different than a primate. But how different is different enough? How different is different enough to make a new species? If there's another bird that looks like that, but it's sort of yellow, is that a new species? Then a turtle that looks just like that, but is 100 times smaller, is that a different species? What defines, ultimately, what a species is? The earliest writings of man, the earliest works that we have out there, the earliest works of science, the ancient philosophers, were all about classifying things. That's like the first thing they decided to do. All of science, they could have started anywhere. They could have talked about what makes fire. They could have talked about why does a wheel turn, and we're just like, these things are different. Because I said so. Let's write that down. Pretty much what they said was a species is an individual, or this is from a translation, a species is an individual organism that represents a group of similar organisms. What are these? Butterflies. They are all one group, so we would say that would be a species because they've got the same form. They represent an entire group. Does that make sense? That's what the ancient philosophers would say. Today, so we've come thousands of years forward, and we define a species as a group of related organisms that maintains a distinctive set of attributes. Those two different definitions aren't very different. There hasn't been much of a change. All of this started with Plato. You don't need to write this down. This is just giving you a historic perspective. Have you guys heard of Plato? Have you read Plato's Republic yet? You need to, because there are certain books that are that you need to read in order to be considered truly educated. The works of William Shakespeare, the Bible, Plato's Republic, Machiavelli's The Prince, a few more. Those are constantly referred to in literary allusions. Really important works to know. Plato's Republic laid out this idea of the allegory of the cave. The ideal form. So in the allegory of the cave, the idea is, imagine, if you will, they're prisoners. They've never seen the light of day, and they're living in a cave. And because somebody's evil, they're forcing the prisoners to look at one wall. So far, so good? These prisoners can only see the wall. Behind them, there's like a puppet show going on in front of a fire. So the shadows are cast on the wall. So what the prisoners think of as real are the shadows on the wall. So far, so good? It's not real, but it's what they conceive of as real. From there, Plato was able to say, there are things that we perceive, what's on the wall, and things that are real, what's going on behind us. There is some real form that allows us to understand the world around us. So ultimately what that led to with Plato, and I hope I don't die with this, but what is this? It is a chair. And you know it's a chair because in your head you have this idea of what a chair is. That's called the ideal form of the chair according to Plato. It's this mystical form that just exists in the nether. It exists only in your mind, or he was thinking more in the spirit of the chair. You know it's a chair. What are these things you're sitting in? Not trying to trick you. They are also chairs. How do you know? Because you got this idea in your mind. What makes a chair to you? It's something you sit in, right? It's defined by its function. What else defines it? Its form, right? Form determines function. What's the form of a chair? 4 legs, a back. All right, so here's the deal. What if This chair had no back to it. Then it's a stool, it's not a chair. Okay, that's cool, it's cool. What if we split back? What if we still have a back to it, but it's got 3 legs? Oh, it's a broken chair, but it's still a chair. So we've defined the chair as something with 4 legs, except when it doesn't have 4 legs, but we still can recognize it as a chair. What if this chair was actually really long? What is it then? Then it's a bench or a couch. But it's the same thing. What differentiates a bench from a chair? Think of a couch and a love seat. You guys know those 2 things, right? What's the difference? Did you just say one's hanging from the ceiling? Okay, so a love seat, for those of you who don't know, it's like a shorter couch. Are you thinking like a hammock? What do you? Kind of. Like a no. Okay. Can you describe? No. Okay. Sorry. Remember, I'm recording this for you for later. So, love seat versus a couch. I'm sorry, I lost my train of thought on that. You know, you have this idea of one. There's a certain point where it goes beyond, and you understand it automatically to be different. That's what the form is. You've got this idea in your head. And so what we did with species was the same thing. We know it's a lot. We know what organism it is, what species it is, because we know we have an ideal form. We know something is a lion because we have an ideal form of a lion in our head, and thus, if that matches up close enough to that picture in our head, we know it's a lion. We know this is a podium because it matches that which is in our head, the form. So those are Platonic forms. One of Plato's students, Aristotle, he ended up deciding to take this even further. It's not just we know it because we know it, which for some people that's good enough. I just know. What he did was he said, we're going to subdivide everything else. We're going to have the genus, the general, and the species, the specific. So Aristotle started out with this broad concept: substance, matter, stuff. And from there, he broke it down. Now the substance, the general, he termed the species. And from there, he broke it down to body and spirit. Well, spirit you couldn't really measure. It's off in the spiritual realm. But there's body, something solid. And then he broke things that are solid down into 2 categories: living and non-living things. Pretty easy. Then from non-living things, he broke that down. Plant and animal. And then he broke that down further. Man and beast, and then stopped. That's the end. Who cares any farther than that? Pretty simple breakdown of how the world works. There's us and everything else. That was the start. That was because then scientists came after him, and they said, you know what? Let's take this further. Let's keep breaking these things down more and more and more specifically. Let's go from the very general to the very specific. What these early scientists and these early philosophers thought was that everything has a vital life essence, something that was distinct and unchanging, and everything that represented that object contained that same essence. What is this? It is a lion. How do you know it's a lion? Right, it's got hair on it. It's got lion face, 4 legs, tail. It's got all of the qualities, the essence of a lion. So what the ancient philosophers would have said was, our vital life essence, or human life essence, recognizes that essence, and thus we know it is a lion. So they began to start classifying everything. There's a group of scientists called taxonomists. Taxonomists, their whole job is to separate things out, to identify them by their characteristics, and then cluster them by those specific characteristics. Early on, it was things like color, locomotion, behavior. Like for animals, an early way of classifying animals said, all right, we got some animals here. These animals have red blood. These animals have no red blood. All right, cool. We got red blood, no red blood. Let's separate out those no red blood. Some of them are hard-bodied, like insects. You guys have squished an insect before, right? Red blood doesn't squirt out of them except mosquitoes, and that's your blood. Sort of this ichor comes out. or soft bodies, slugs, stuff like that. And then you went down with soft bodies and you said, okay, well, there are some soft body things that live in shells and some soft body things that don't live in shells. Classifying by breaking it down smaller and smaller and smaller. Now, these are really, what's the word I'm looking for? Really sort of haphazard classifications. Who would have thought, let's just look at animals and classify by red blood or no red blood? How else might you have classified them? How would you personally have taken this big group of animals and split them apart? Sure, things with bones, things with no bones. How else might you have done it? Size, big things, little things. How do they move? This thing flies, this thing swims. Any of those are just as good of an answer as the others. It's just our innate need to classify things. Now, speciation becomes significant in the modern world primarily because of business models. In modern molecular biology, you can trademark and patent organisms, but only a species. You can't just patent a broad range, a genus, the general. If you were to create, and it's very possible that you do this, you were to create a microorganism that does something cool, maybe breaks down, maybe it's yeast that breaks down ethanol faster, or sugar faster to create ethanol. You make a super yeast that helps ferment things to make an awesome, awesome alcoholic beverage. You can patent that, sell it, it's yours, you make money off of it. If somebody else comes along and changes it just enough to where it's a new species, they can now patent it. Does that make sense? So speciation becomes important for economics with modern patenting and trademark laws. Okay, now we're getting to the meat where you're going to have to really start writing things down. Each species is set apart by a distinct set of characteristics. All species are separated out. Now, it's up to the taxonomists to decide which of these is most important, but these are the general ways that they set things apart. They're morphological traits. What do they look like? Reproductive isolation. Can they mate with each other or not? Molecular features. Are they genetically similar? Are they genetically different? Ecological factors. Where do they live? And their evolutionary relationships. How closely related are two things based on what their ancestors look like? If we're looking at purely morphological traits, you know, the stuff that we see around us. Morphology is what it actually looks like. This is how the earliest taxonomists classified everything. That's all they knew. What does it look like? Up there in the top corner, what are those? Not trying to trick you. Yes, they are fish. How would you classify those fish? By color, right? Or by size. I mean, you've got to figure out what is their top level. First, color. Red ones, yellow ones, black and white ones, assorted other. Then from there, it breaks down even more. You might go with fin shape. You might go with size. You might go with anything, really. But you've got one level that you as the taxonomist have decided that is the most important. And then you break it down farther into the next most important until finally you've sorted everything out. These are angry birds, right? How would you sort them? Which one, there's the general group of angry birds. Which one's the most different? Why is the one over on the far right, left? Okay, that one's a triangle, the other one's a round. That works. How else what's the next level? You looked at shape first. What would you do next? Color? They're all different colors. You separate them out. What would be the next one you do? Long legs versus short legs. Their beak shape. Color would be the final thing, probably the lowest level. You got all sorts of things. You, as the taxonomist, would end up placing your own values on what is most important to show relationships between these organisms. So that's morphological traits, the physical traits. You might look at reproductive isolation. Establish nature of population based on the ability of that population to interbreed. That is, procreate and produce fertile offspring. If you can procreate, then you are the same species. If you cannot, you're a different species. Does that make sense? If you can mate with something and produce fertile offspring, you are a unique species. If you get mate with something and you cannot produce offspring, you are not. Well, so say that again. Well, right. So what about mules? Mules are a horse and a donkey, right? So you got a horse, you got a donkey, you breed them together. So are donkeys their own species then? Are they not any species? Are they just biological waste product? Don't know. Quite harsh. Here's this is a cross between a donkey and a zebra. We've talked about this before, right? It's not a zonkey. It's a zebroid. It should be a zonkey. I swear we talked. Katie, did we talk about that? You still think they're zonkeys? I swear they're zebroids. But Katie says. This is not viable. Is it its own species? Is it neither a horse nor a zebra? In the end, doesn't matter. This is a crocoduck. No, it's not a real thing. It's a real thing. That'd be horrible. That is a real thing. This is not a real thing. That'd be terrifying, wouldn't it? Crocoducks. If you took a crocodile and a duck and tried to mate them together, comedy happens. But would there be a viable offspring produced? No. You'd either have the duck would be dead no matter what. I mean, let's just be honest. The dead duck comes out of that. I mean, whether the crocodile decides to eat the duck or actually tries to mate with the duck is dead. It's not going to happen. These two populations don't mate. You can look at distinctive molecular features. You look at how closely related two organisms are by the percent to which they share genes. Humans and chimpanzees are less than 3% different when it comes to their genes. 3%. Gorillas and chimpanzees and humans are 2% off. So you've got you've got to figure out exactly what percentage makes you human. Oh, and by the way, humans are actually slightly different from each other in about, I think we have about 2 to 2.5% differences between each other. So there could be a greater difference between you and another human genetically than there is between you and a chimpanzee. And that just probably answers a lot of your questions about, like, your family. Kind of neat. The tricky part with molecular features is determining what are the critical genes that make you human. What are the critical genes that make a chimpanzee a chimpanzee? So this is a newer field. Ecological factors. The nature of a population can be established based on what type of environment the group thrives in. Where do they forage for food? Where do they prefer to mate? Where do they prefer to live? Their ecology. What they do, how they do it. You've got two broad types of primates. You've got tree-dwelling primates and ground-dwelling primates. You could separate out their species by saying, you live in the trees, you live on the ground. Different locomotion techniques. These guys tend to swing and move on all fours. The ground-dwelling ones tend to walk on two legs. You've got two distinct ecologies that you can draw from, representing two broad groups. And then you could break it down. And then you can look at evolutionary relationships. The nature is based on fossil evidence that ultimately led to the population of interest, or by establishing evolutionary relations of populations through gene sequencing. So if you were to look at specific organisms and the fossil record, try to figure out how they're related to each other based on transitional fossils. Yes? For ecological facts, why wouldn't you do ocean and land? Yeah, no, that would be a great one. That's a very broad original split. This lives in the ocean, this lives on land. That's a great way to start off. That lives in the sky. So is tree dwelling or around dwelling like this? Yes. Oh, yeah, no. I'm just giving you broad, examples of. Sorry. What I'm trying to get across was where they live. So those are your 5. Bear in mind, with those 5 distinctions, it is ultimately up to not nature to figure out how these things are related, but the taxonomist. And the taxonomist is the one who places their own personal biases, and there are biases, their own personal beliefs, into what is the most important. Yes? I don't know the answer to the question. Did the question? So she asked, did we ever answer the question on if they can't reproduce? Are they a different species or not? Are they a separate species? Do they have their own species? Don't know. They're given a name. Therefore, they're their own species, right? Because we're humans and we like to give things names. I named my pets. If you spay a cat, are they suddenly no longer a cat? They can't reproduce. We've got Alfred Wallace and Charles Darwin. These are the guys who came up with that concept of natural selection. And they came up with it at about the same time. Whenever you hear evolution, you always hear these guys. Pretty much they're the same in that they came up with evolution, natural selection, and they have awesome beards. They were really interested in the species concept. How do we define life? How do we distinguish one group of organisms from another group of organisms? That's the big thing. And we're still doing that. What makes a species? What makes things different? And right now you're starting to ask yourself, and I swear you have to be because you're in this class, you're starting to ask yourself, wait, okay, I thought I knew what a species was, but it turns out there are a lot of problems with everything. We're going to get to even more problems. Our current scientific understanding of species says there is absolutely no rigorous, comprehensive way to distinguish between species in all cases. So we as biologists are trying to figure out a way to operationalize this, to make it so that we have an operational definition of a species, even if it's not perfect. Does that make sense? We know we can't have a perfect definition. And that's painful for scientists to say. But the reason we can't have a perfect definition is because a species is a human concept. It's a human construct. It's not something that nature rigorously defines. Does that make sense? A zebroid doesn't care whether we call it a zebroid or not. It might think it's a zebra. It might think it's a donkey. It might just think, God, I want to eat some more hay. And try to reproduce. That's what I imagine goes through a zebroid's head. Here, this bottom picture. This is a tigon. This is a tigon. A tigon is a cross between a lion and a tiger. Now, yeah, I know, you can do that. One of the biggest of the big cats. It depends on which direction you're going, which parent it is. There are ligons and, there are tigons and ligers. I forget which parent is which, but it all depends on which is if the male is the lion or the male is the tiger. Why does it matter? Yeah, it comes down to this one has a mane to it, and the other one doesn't have a mane to it. It's a sleek look. So, yeah, tigons and ligers. They're huge. Are they their own species? Well, they can reproduce. If you took two tigons and put them together, they could reproduce. If you took a tigon and a tigon and a tiger and put them together, they can make tigers. If you took a ligon and a lion, they can make lions. If you took a tigon, sorry, this is getting confusing in my head, a tigon and a lion and put them together, they make baby lions. Yeah? Tigon? Yeah. A lion. Or it would make a mix. It depends on what you as the taxonomist think. Does that make sense? This is also subjective. Get this. All right. prokaryotes, right? Unicellular organisms. We can look at those and say, this is a paramecium. That is a species, right? How do we know that is a species? That is, it doesn't mate with anything else, so you can't define it by mating. It just makes its own, it splits in half. Why is it that each and every individual paramecium that splits itself in half is not its own unique species? Because that's exactly it, because we didn't say so. Because that would make it too hard. There are 4 competing species concepts. Poor Amelia just left. And I'll tell you, somebody, and I'm not going to mention any names, but somebody's going to ask, describe the 4 competing species concept, and then describe which one is your favorite and why. The biological species concept, the evolutionary lineage species concept, the ecological species concept, and the general lineage concept. So first up, the biological species concept. If 2 creatures cannot reproduce to produce fertile offspring, then they are different species. Makes sense. You cannot reproduce with a gorilla, 2 different species. A dog cannot reproduce with a cat, different species. Does that make sense? And that's probably the most accepted definition. It's very functional. It doesn't work for prokaryotes or for unicellular organisms, though, that reproduce asexually. And then you've got those issues with, like, the horse and the donkey, right? You don't know if a mule's its own species or not. This is kind of cool. Did you guys know that whales and dolphins can breed? Well, yeah, and they're like, no, don't buy it. True. Don't forget there are small whales. That. Sorry. If you were to take a dolphin and a pilot whale and cross them together, you end up with a cetacean. It's definitely a whale or a dolphin. Don't know which. That looks nice, yeah. And it's fertile. What species is it? Don't know. But it's definitely something. That's a plant, right? Daylily. This is a triploid daylily. It reproduces through, the only way it reproduces is through vegetative growth. That is, stuff grows off of the sides. It gets cut up, and that can be distributed. It's almost like, you guys ever grown strawberry plants? Or you see the strawberry plant will sort of put out this really long runner, and it grows a whole new plant on the end? Or a spider plant does that too? This does not make seeds. It doesn't reproduce sexually. It doesn't get crossed with another lily in order to make new daylily seeds. Therefore, it's unique. Does that make it its own species? Or is it related to its parents? Its tetraploid parent or its diploid parent? Its 4N chromosome parent or 2N chromosome parent? Very different from each other. One has twice the number of chromosomes as the other, and they produce an offspring. So then, is this offspring going to be the mother parent, the mother parent, the father parent, or is it going to be its own unique species? Right now, science is siding toward this is a unique. The daylily is unique. The hybrid whale is not. Then we've got the evolutionary lineage concept. Here we define lineages by separate evolutionary pathways. Each species is a direct descendant of an immediate ancestral species. So you are human, right? And you know you're human because what were your parents? Human. And their parents? Human. And their parents? Human. And then we get to a point where you don't know for sure. Oh, I don't know. Maybe, do you know your great-grandparents? Okay, fine. Let's just keep going back then. Another 15 generations, say. You don't know them. We don't know for sure, but we do know that over time there are slight changes. We know that 15,000 years ago, humans were very different than they are now. We may not even really be able to call them Homo sapien. They might have been Homo florensis. They might have been another offshoot.
Was it this class that had the conversation where people were saying that we're now called Homo sapiens sapiens? Like
it might be. I have no idea.
Someone told me that we're Homo sapiens sapiens.
I don't think that we've talked about anything like that. I'll talk about renaming geologic ages later. But the idea is, you know that two species are different if they descended from the same common ancestor. You know what that common ancestor was, or you have evidence that it had an intermediary form, and they are both different from that common ancestor. So there's a split. The problem with this is you need to know those common ancestors. You also need to figure out how different is different enough. At what point do you say, this is a species, this is a different species? Because it's almost moving too slow to tell. It's that whole idea of boiling a frog. Do you guys know this? Have you heard of boiling a frog? Don't boil a frog. That's evil. But if you were to boil a frog, you take a frog, you put it in a pan of water. This is not an experiment we're going to try in here, because frogs are expensive. You take a frog, you put it in a pot of water, and then you turn the water on the stove. The frog will keep swimming around in it. As the water temperature increases, the frog doesn't leave because it's moving too slowly for it to, the temperature changes are moving too slowly for it to realize that it's dying. So over time, the frog will just stay in the water and die because it doesn't, because the pace of change is so slow. Similarly, for us, at what point do you suddenly go from being Homo neanderthalis, or not even Homo neanderthalis, I don't know, one of the earlier Homo species, to being Homo sapien? Where's the shift? Where is it where somebody's parents are this earlier form and they're the new one? And I know every generation thinks that, 'cause they're like, yeah, I'm way better than my parents. Way smarter, way faster, things like that. Where's that evolutionary jump? So that's the evolutionary lineage concept. You've got the ecological species concept. You define an organism as a group sharing a common ecological niche. Usually that's a unique set of environmental conditions. Let's say, what are they? Not wasps. Yellow jackets. You know yellow jackets, right? Does anybody like yellow jackets? No, nobody likes yellow jackets. Yellow jackets are horrible, horrible creatures, and we all hate them. They don't even do much pollinating. I mean, they kind of suck as far as organisms go. Yellow jackets will sting you. They live in two areas. Sometimes they live up in the trees. They live in these hives that they formed with mud and with their spit. You see them, you avoid those. You know that. More often, we encounter them when we're cutting the grass, and we discover an underground nest. These are the same general group. They can interbreed. They can produce fertile offspring. Some prefer, though, to mate high. Some prefer to mate low. This, in some taxonomists' mind, this ecological split is what would determine whether or not one is a species compared to the other, or if they're the same species. So you're looking at several species concepts here. And it's going to be up to you, the taxonomist, to figure out which one you like best. You've got the biological species concept. If they can mate, they're the same species. These guys can mate, they're the same species. The evolutionary lineage concept. If they have a common ancestor, then they're going to be the same species. Well, these guys definitely have common ancestors. Same species. Or ecological. They live in different places. They eat different things. They tend to breed in different places. So they're different species. Each concept might call these a different, whether a species or not. And the final concept is the general lineage concept. This is the one I think your book uses. It says each species is a population of independently evolving lineages that evolved from a specific series of ancestors, and as a consequence forms a group of organisms with a particular set of characteristics. That is a long, convoluted answer. What it's saying is it tries to combine that evolutionary lineage concept. Each species is independently evolving from a set of common ancestors. Again, you would pick up the problem with the evolutionary lineage concept, which is we don't know what that common ancestor is. And it forms a group of organisms with a particular set of characteristics. Well, where have we heard that before? Group of organisms with a set of characteristics. The exact same thing we heard 2,000, 3,000 years ago. We're right back to where we started. So here are 4 concepts, species concepts. Any of them are considered acceptable. But what we need to decide for in here is which one is the best. Which one is ideal? Which one do we want to use as our operational definition? So, what I want you guys to do is spend the next few minutes discussing these 4. I don't know, talking groups of 3. And then we'll bring those groups of 3 together. I want you to discuss these 4 concepts and figure out what operational general species concept we are going to use in this class. And I will base the rest of the lectures on what you guys come up with as how you want to define a species. To it. All right, so after much discussion, it looks like we're going with the biological species concept, which is good because that's what I based this lecture on. When it comes down to biological species, you've got 2 types of what's termed of isolation. Prezygotic isolation and postzygotic isolation. What does pre mean? Before, and post, after. So far, so good. Zygotic. That's a little bit harder. The zygote forms. When does the zygote form? 2 gametes come together and form a zygote, right? So prezygotic is before fertilization. Postzygotic is after fertilization. If you were to have prezygotic sexual isolation, that is isolating yourself sexually before mating. It's just not going to happen. The sea slug is one of my favorite examples of prezygotic isolation. Sea slugs mate in gigantic sea slug orgies. They are nuts. And the reason they go crazy all at the same time is because a pheromone is released that gets amplified in the water, and it causes all of the sea slugs to suddenly want to mate. Now, if a sea slug had a mutation and it was not able to receive the pheromone, suddenly everybody else is ready to mate except for that guy. That means he's left as like, what's going on, guys? Why are you all doing this? I'm just over here eating. No mating. The common field cricket. Crickets chirp, and they chirp to attract a mate. These chirps are at a specific frequency per species. So, if one is chirping at a particular frequency, it's able to attract a mate. If it gets a mutation, and it cannot chirp that high, its hoarse, well, not hoarse, its legs aren't working very well. It's not able to run them against each other. Suddenly, it can't make the same sound, so it can't mate. These are prezygotic mechanisms. Yes?
Do you know what that little thing at the butt of a cricket is? Like the black thing coming out.
This thing right here?
Yeah.
I am not sure off the top of my head. I know in flies they're they're called sex combs, but those are I'm not sure what it is in crickets. Nope, crickets won't sting you. So these isolating mechanisms happen before insemination. This is a mating dance of a bird. If they get the dance right, they're allowed to mate. If they get the dance wrong, they don't get to mate. These dances, these displays are genetically determined. So a slight change in your genetics, a slight change in your behavior means you're not able to mate. There are 5 ways of isolating yourself prezygotically. Habitat isolation, temporal isolation, behavioral isolation, mechanical isolation, and finally gametic isolation. We're going to go over each. First off, habitat isolation. If a geographic barrier makes interbreeding between populations impossible, you are isolated in your habitat. To summarize, if you are not in the same location as the person you want to mate with, you don't get to mate with them successfully. You could try, it's not going to work. So habitat isolation, geographic isolation, is where you are separated, or a population is separated by a barrier. They're separated by some commonality. They're separated where one particular group of the organisms prefers to mate in one place, another prefers a different place. Here's an example of habitat isolation. You've got two groups of flies. Originally, they're a single population. You split them apart. Some of them are only fed with maltose. Now, in case you didn't know, flies like to breed where they eat. So pretty much, they like some maltose with some ethanol, give them some alcohol, and these flies like to mate. The other set of flies, you just gave starch and ethanol. Over many successive generations, these flies separate out to where the maltose-loving flies will only mate on maltose. The starch flies will only mate on starch. So when you put them into a container together, they're not going to be able to mate with each other. The starch ones mate on starch. The maltose ones will only mate on the maltose in that container. It's like two entirely different. Okay, how about this? It's like you've got two places where people might meet in order to mate. Not in order to mate, but they might meet and then mate later. You've got a museum or a library, and you've got a bar. You're going to have one group of people with a specific set of characteristics at the library and another at the bar. They're going to end up mating. Their offspring come out. They will then pass on their particular preferences, or those offspring will pick up some of those preferences. And then that will be reinforced over and over and over again. That's isolation through habitat. Here we got another one. You got a single population, a river runs through it. Those populations are separated and cannot mate together until eventually they are two totally separate populations. You may end up with temporal isolation. Due to the timing of the mating cycle, you may not be able to get interbreeding between two populations. They're isolated through time. You cannot mate with, I don't know, Abraham Lincoln. Frogs in spring and frogs in fall. Some frogs will mate together, even in the same pond. If they're not on that same mating cycle, they're not going to be able to be a interbreeding population. Cicadas. You guys have heard cicadas around here before, right? I know it's not as bad down in southern Virginia. Up in northern Virginia, it's really bad when they come out. It sounds like almost like alien laser cannons going off all the time. There are these guys operate in cycles. There's the 1-year cicada, the 3-year cicada, the 5-year cicada, the 7 and the 17. Some years, they all pop out at the same time. Those are sort of these apex years where it's really loud out. But most times, you're going to get just one batch popping out. And that batch pops out, they come out as these sort of grubs. I don't know if you've seen them before. They look like big, thick white worms. They come out, they form a, they molt a little, they end up having, going and removing their exoskeleton, getting these big wings, fly around. When the cicada finally gets its flying form, have you guys ever seen these before? When they get that flying form, they no longer eat. They don't eat, they don't drink, they don't do anything. Their whole job is mate. They can last for 1 or 2 days, and then they die. The eggs they lay hatch underground. They live for 17 years underground, and they come out for a day or 2. Temporal isolation. Those at the 5-year very rarely end up with the 17-year. Crickets. Some crickets mate in fall. Some crickets mate in summer. They don't overlap. So you've got, even in the same field, you'll have different populations of crickets. You might end up with behavioral isolation. This is one that will end up happening a lot with humans. Different populations have different ways of signaling mate choice to their prospective mate. To be able to signal, here is how, or here I'm ready to mate, or I do not want to mate. That can be especially dangerous in the animal kingdom, where a signal for I'm ready to mate and a signal for get off of my territory are very similar. Birdsong. Birdsong does two things. It says, I'm ready to mate. And it also says, I'm going to kill you if you don't leave this area. If one bird can't interpret that, suddenly he's not only left out of the mating cycle, but he's going to try and mate with somebody who really doesn't want to mate. Does that make sense? This is behavioral isolation, these behaviors. Yes, birdsong is genetic, but it is still a behavior. Insects. This is fruit flies. I have done a lot of research on fruit fly mating. Way too much. Fruit flies have a very specific mating dance that involves the movement of its wings, the exact frequency of the humming it makes. If a male fly does not do things absolutely right, the female will reject the male, and he is left out of the mating pool. Now, if there's a mutation in the female, and she doesn't recognize the male's mating dance, she's going to reject him anyway. She's out of the gene pool. If it happens to the male, he's out. If, though, two of them happen to find each other, two that happen to be a little bit different than the rest, and you've all been in that situation, you found that person who is just a little bit different from the rest, just like you are, a couple of black sheep finding another black sheep, yeah. Things work out. They end up being able to produce a new species. That's how speciation happens. For insects, if they don't line up just right, so what happens is the male comes in, and he orients himself. First he comes in from the side, so she sees him because they got some big eyes. Sees him, and then he goes around the front. Then he goes around the front. He's like, hey, check out my eyes. He sings her a song, and by sings, I mean he flaps his wings very quickly. That vibration will trigger the female to walk away. Now, for humans, that's like, she's leaving, let her go. For flies, that's an invitation. So he follows her. Then he licks her, because that's very important for flies. Then there's a copulation event, and then she ultimately rejects him and he leaves. It has to follow the same pattern every single time or it doesn't work. In humans, eye contact can mean lots of different things in lots of different cultures. Eye contact in America could mean, are you listening? Do people make eye contact to find out what's going on? Do you see what I'm saying? In other cultures, eye contact is flirtation. In cultures other than that, eye contact is it's a challenge. You're holding eye contact. That means you're challenging me for dominance. Can you... I don't know how I can put this. It's amazing that we are able to live as a species together when our cultures, our behaviors are so different. And that leads to misunderstandings that can lead to violence or relationships. One of the two. Does that make sense? So small changes in behavior will lead to sexual isolation. Mechanical isolation. This is a fun one. Morphological features of the organism prevent the two species from interbreeding. The parts don't line up. Here's an example of mechanical isolation. These two dogs, it is unlikely that they will successfully mate with each other without assistance. I guess the small one might use a stepladder. But that's an isolation example. Here's another one. These are copepods. The plates on one species don't match up to the plates on the other, so the genitalia never line up. They cannot mate. Mechanically, they're isolated. And then finally, we have gametic isolation. This is where all of those behaviors worked, all of the parts lined up, you had copulation. The gametes are coming right at each other. They're rushing toward each other. And then it doesn't work. The zygote never successfully forms. So, what we're looking at here are sea urchins. Yeah, sea urchins. Sorry, that's double checking. Sea urchins are going to release their gametes all at once. You can entice a sea urchin to release its gametes. Here is if you ever want to get a sea urchin to release its sperm, here's what you do. Because I know. What you do is you take a needle full of saline and you inject it. And what that needle full of saline does is it triggers it to say, to just release all of its gametes all at once. Does it with males, does it with females. In this way, you can collect a whole bunch of sperm, and you can collect a whole bunch of eggs. We used to do this as an experiment at University of North Carolina, Greensboro, so that we could look at meiosis. In here, we have this sort of paper lab where you guys just... Here, we actually took sperm and eggs, put them together, and saw what happened. There are a lot of sea urchins in the ocean. They're all releasing these gametes. Those gametes have to find each other and unite. But what happens when a gamete from one organism meets a gamete from different species? They sort of just bounce off each other. They don't fuse. They don't have the right protein-protein interactions. So that's gametic isolation, where those sperm are not able to fertilize the egg. And that gives us all of our examples of prezygotic sexual isolation. All right, so while prezygotic isolating mechanisms happen before the zygote forms, postzygotic happens after the zygote forms. So at this point, you've had an egg and a sperm. They've come together and they've formed an offspring. Take it back. They've formed a zygote, a developing organism. There are two big hypotheses surrounding postzygotic sexual isolation. One of them is known as Haldane's rule. This is where when you have an F1 offspring, so if you have two dissimilar organisms mating, two different animal races, what you'll see is one sex becomes absent first. And if that sex becomes absent first, it's usually the heterogametic sex. So in humans, what's the heterogametic sex? Which sex has two different sex chromosomes? The what? The male. The male has an X and a Y. It threw me for a second, because you usually get things right. I was going to go with you on it, too. I was like, yeah, this is a female. So in the F1, if there's an issue with the offspring, usually the issue is the male. The female's usually fine because the other X will compensate. That's known as the large X effect. So Haldane's rule can be supplemented by the large X effect, where if either of the two genders are sterile, it's going to be the male that becomes sterile first. It all comes down to that X and Y chromosome. If there's a problem on the X chromosome, the Y chromosome won't be able to compensate for it. That might lead to hybrid inviability. Here you have fertilized eggs, so an egg and a sperm were able to fuse. You got through all that behavioral issue, you were in the same place at the same time, you overcame all the prezygotic isolating measures. When you finally mated and produced a zygote, it doesn't develop beyond very early development. So what happens is you have the egg and the sperm fuse, the zygote then splits into two cells that are held together, 4, 8, 16, 32, 64, 128. And sometime in that early cycling, the cells begin to just stop developing. They stop forming. They stop undergoing mitosis. They enter into a permanent G0 state, and the organism is never born. It's never fully developed. that's called hybrid inviability. The zygote is formed, but the offspring is never born. That's one type of postzygotic isolation. Another type is hybrid sterility, where you have a viable species, two viable species, two species that can produce offspring. Those offspring may end up becoming sterile. So you have two very similar species, a horse and a donkey. If they were to mate, they might produce an offspring that is born and grows up. But that offspring may not be able to successfully reproduce with other organisms due to polyploidy. It has either too many chromosomes to mate with one of its parents or too few to mate with the other. So there are two big kinds of postzygotic isolating mechanisms there, hybrid inviability and hybrid sterility. And whenever these come into play, which ones will happen, what gender will it affect the first? The males, due to the large X effect, or Haldane's rule. It's been theorized that prezygotic sexual isolation evolves much faster in a population than postzygotic sexual isolation. Prezygotic isolation is all about behaviors. It's all about location, things that you can shift very quickly. Postzygotic isolation occurs relatively slowly because that's going to entail molecular changes. You have a distinct, quick change to a reproductive gene. It's far easier to change behaviors than it is to change your genes. Does that make sense? For birds, they're more likely to change their mating dance than they are to be able to change the capability of their gametes from being able to fuse. So there's a connection. After contact between formerly separate populations, prezygotic isolation and postzygotic isolation can arise. So if you take two populations and you split them apart, over time, small differences in those populations can keep increasing. So they become more and more different. You've got these two totally separate populations. You bring them back together, and differences have arisen. Think if you were to take groups of people. You take one group of people, you move them over by the coast. You take another group, you move them over by the mountains. You leave them there for 200 years. No contact between them. Will differences arise? Yes. They'll have different values, different skill sets. They'll look different. They'll talk different. They may have invented new words that the other ones will never have seen. Behavioral differences arise very quickly. Will those two populations still be able to physically mate? Yes. And still produce fertile offspring. Will they be attracted to each other? That's a different question. It's more likely for those prezygotic isolating measures to develop first. They come around earlier than the postzygotic ones. Once you've gotten those differences in prezygotic isolation, once they no longer mate together, then suddenly, not suddenly, but over time, those unique populations will develop postzygotic isolating measures. It's sort of an after the fact usually. So prezygotic isolating measures usually happen first. Prezygotic isolation is probably the most predominant way that we get speciation. They reinforce species boundaries. And they're probably a necessary step to dividing one species into two. What we're looking at here is what are known as ring species. Each of these is a separate species. And this is going to be weird. We'll get into this in a few minutes in the next lecture. But the idea is this lizard in the middle here cannot mate with the one right below it, can't mate with the one right on top of it. They don't. They have prezygotic isolating mechanisms that stop that. But the middle one over here and this one in the bottom right, they can mate and reproduce. They produce fertile offspring. There are prezygotic measures that reinforce this species difference, but not that one. because these two species never run into each other normally. They run into each other in zoos. That's about it. So prezygotic can help divide a population into large species. This fits very nicely with that biological species concept. Because typically what will happen is the hybrids will have a reduced fitness. The lower the fitness, the lower the W value, the less likely it is for hybrids to survive. That is, the two separate species, or the growing separate species, will have a greater fitness level and pass their alleles on to their offspring with each other, those homospecific crosses. So viable hybrids, as long as hybrid viability decreases, as long as you lose fitness in the hybrid, you get species reinforcement. Reinforcement of mating isolation can happen when you've got these two separate populations coming back together. If you've got that prezygotic isolation, that the two populations choose not to mate with each other or can't recognize each other's mating signals, that's going to happen first, and that will also reinforce speciation. The big thing for speciation, big takeaway message, is we have to take one population and somehow split it into two, and then leave those two populations over time to develop enough differences that they either choose not to mate with each other or cannot mate with each other.